Welcome back to Polymeric Biomaterials. We have been looking at biomaterial performance, and under that theme, this is the last lecture, and it also happens to be the last lecture for this course. And this lecture is titled Biocompatibility, Medical Devices, Translation and Regulation. Now, at face value, it might seem like these are just a bunch of words that have been strewn together, but I assure you that by the end of this lecture, you will see a very deep connection between these and why we are looking at them together. Essentially, what we will aim to do in this lecture is to connect all the dots for you from various angles and concepts that we have seen throughout the course, both from a biomaterial perspective and a host perspective. So that will help explain how biocompatibility can be contextualized for medical devices and also the aspects that need to be kept in mind when you want to translate, and what kind of aspects do regulatory agencies look at when we want to translate medical devices. So, with that quick introduction, let's jump right into the contents for today's lecture. We'll first start off with a quick look at devices, implants and translation, which we have already looked at in previous lectures. Then we will focus quite a bit on testing, particularly biological testing. We have previously seen testing before, but that was more mechanical testing, and we looked at bulk properties and responses. Here we are going to be focusing on biological testing, particularly with regard to biocompatibility and blood compatibility, as well as toxicity and sensitization. And then we will take a look at immune response to natural materials, which we have not looked at in any detail in previous lectures. So, we will focus on that a little bit and look at the risks involved in implanting natural materials. And then we will turn our attention to potential adverse responses that biomaterial implantation can have, and what kind of lessons can we learn from retrieval. And then we will move our attention to a couple of very important case studies. One of them is the darkened shield IOD case study, which is an intrauterine device. The other is the Vitek implant case study, which again we have looked at in previous lectures. And then that will help us segue towards why regulation is required for medical devices, and what kinds of aspects do agencies such as the FDA look at as far as regulating medical devices. And then we will conclude this lecture and the course with some closing remarks about biocompatibility and the use of biomaterials in medical devices. So, with that introduction, let's first start off by looking again at biomedical devices and implants, and this would be a good time to pause and recall that we have already seen an entire lecture, lecture number 4, connected to devices. This was biomaterials from a device perspective in one of the early lectures in this course. And we have also looked quite a bit at the use of synthetic polymers, which is what is shown here on the slide as examples. The use of synthetic polymers in such devices we have dealt with in great detail in lectures 17 and 18. And shown here are just 3 examples of that. One is a drug-eluting stent, the other is a total knee replacement device, the third is a breast implant, all of which use synthetic polymers. For example, the drug-eluting stent has a proprietary polymer coating that releases a drug to prevent restenosis. The total knee replacement, as you might recall, has an ultra-high molecular weight polyethylene liner that's used as part of the device. And of course, the breast implant has silicone based materials. Now, looking at medical implants and devices, it will be good for us to look at formal definitions of what they are. They are essentially intended for use in the diagnosis, prevention, treatment and cure as well of diseases or other conditions. They can consist of processed tissues or synthetic materials, although we are looking at synthetic materials here, they can also consist of processed tissues. But one key aspect to keep in mind is they are intended to affect the structure and function of the body without chemical or metabolic action. That's a very important distinction that separates medical devices from medicinal products. So that's good to pause and reflect on here. What we are focusing on is medical devices, and it turns out that this distinction is quite important, although it might seem trivial. It's quite important from a regulatory agency perspective. What regulatory agencies look for in terms of a device versus a medicinal product are quite different, and they are also governed and regulated by different wings of the FDA, for example. Medicinal products will include, it's an umbrella term that includes drugs, biologics and so on, immunotherapies, all of these things. Those are also important, but they are not part of the medical device category that we are talking about here. Because the key difference here is the medical devices, as per this definition, is primarily going to serve a physical or a mechanical function largely intended to affect the structure and function of the body. Whereas a medicinal product such as a drug or a biologic is going to exert its efforts through biochemical means or pharmacological, of course, or immunological. So that's what we mean here when we say chemical or metabolic action. So the medical device itself will not act this way. At least the primary intent is not this. Now you can see from this perspective, the total knee replacement and the breast implant serve or fit these definitions, where largely they are serving a mechanical or a physical function, and they serve to affect or enhance the structure and function of various body parts. But the drug-eluting stent is an interesting case in point, because we have listed it broadly under the category of medical devices, and yet you see that there is a drug involved. So it turns out that the primary intent of the drug-eluting stent is that of a medical device, with the drug serving a secondary function. So this is still regulated as a medical device-led product, with of course inputs from the arm of the FDA, for example, that regulates drugs and biologics. So these are some subtle points to keep in mind, but because we are interested now in looking at devices, regulation and translation, we are making this distinction very carefully. Now from a translational perspective, one can ask, what does the bench to bedside pathway look like? Needless to add, this is not a list that we are seeing for the first time. We have seen this list before. I am just recapitulating some key aspects of this for the purposes of this lecture. Firstly, we note that there is a need and a market driven by disease burden or accidents. And then there is the idea of a device design tailored for a specific need, right? So these two go together. Once the need and market and the device design have been identified, or the requirements for the device have been identified, then material synthesis is important, where you are interested both in availability and production of the materials involved, and then you are interested in material testing. Mechanical testing is something we have already seen. Toxicity and biostability testing, which we have started recently looking at in the last several lectures, and we will again look at in this lecture as well. And then there is fabrication, which involves material integration. Essentially, we have seen that biomaterials or polymeric biomaterials particularly are very rarely used on their own as a device. They are usually integrated as part of a medical device, which involves other types of materials as well. So the total knee replacement, for example, is not just the ultra high molecular weight polyethylene liner. It serves as a liner as part of a metallic device, right? So when we look at fabrication, it also involves the integration of different kinds of materials, and of course sizing and shaping based on patient needs and how that can be manufactured at scale. Then there is device testing, particularly looking at safety and effectiveness, both in vitro and animal models. And then comes the regulatory aspects, where you get approvals for clinical trials and follow-up as well. And finally, clinical deployment and use, which is typically patient-centric. And in some cases, unfortunately, you may have to retrieve the implant, either because the implant has failed completely, or perhaps it has undergone wear and degradation and needs to be replaced. So, for all such cases, retrieval is often required, but then what lessons can we learn after retrieving is very important, and that is something we will look at in this lecture again. Now, you see, this is a very complex pathway requiring the inputs from several people, and just one quick list will tell you how complex this pathway is. Engineers, scientists, clinicians, regulatory agencies. So, this is a very short list, because within engineers you can have multiple subtypes, within scientists you can have multiple subtypes. Again, clinicians have different specializations. So, you see that this bench to bedside pathway needs inputs from many different types of people working together to finally develop a medical device that can be implanted in patients. Now, keep this in mind as we go through the rest of this lecture, because this pathway is quite complex. Now, we will first begin by looking at testing, as we have already hinted at, from a host perspective. Now, this testing that we are looking at here is biological testing. So, that is the qualifier we are adding here, it is biological testing, but again from a host perspective. It is going to involve the implantation of a biomaterial eventually, and one thing you can ask is what happens to the biomaterial once it is implanted, both from a safety perspective and from a effectiveness perspective. These are essentially the two pillars upon which agencies such as the FDA will evaluate, approve and regulate medical devices. So, it is important to keep this in mind as we look at testing. We are going to look at what the material does to the body. We are equally interested in what the body does to the material. So, in testing, we first start off by looking at biocompatibility indicators, and this is something that we have started looking at in the last several lectures. This includes blood coagulation and thrombosis, which we have looked at in lecture number 32. The inflammatory response and host response, which we looked at in the previous lecture, lecture number 33. We have hinted at infections and the use of antifouling materials for preventing biofilm formation in the lecture on protein adsorption, lecture number 31. You also have to keep in mind that some of these can induce tumors, which is also part of biocompatibility testing. And of course, toxicity and sensitivity or sensitization, which we are looking at in this lecture on this slide. So, all of this is contained within that broad term that we call biocompatibility. So, that is one of the reasons why this lecture is also titled Biocompatibility in Medical Devices Translation and Regulation, because throughout this course we have looked at several different dimensions to biocompatibility. Now, this is again biocompatibility from a host perspective. Of course, blood compatibility, which is one very important aspect of biocompatibility, typically involves looking at thrombosis or excessive clot formation, perhaps even emboli that can get dislodged, and typically anticoagulant therapy is often needed in most cases to prevent adverse effects of thrombus. Typically in the initial stages, at least after implantation, this is required. In some cases, for example, if mechanical heart valves are used in young patients, this anticoagulant therapy may be required for very long time periods, and that can have its own adverse effects in the long run. The other aspect to testing includes toxicity testing. Now, toxicity can be of different types, and we have just listed some aspects here just to give you a taste of what toxicity testing involves. The first one is systemic toxicity, which is you are going to implant biomedical device in a certain region or site in the body. But what you're interested in looking at in systemic toxicity is toxicity away from the site. Essentially, does this cause toxic effects away from the site systemically, particularly in organs such as the kidney and the liver? Here the route, duration and frequency are important, and you are interested not just in acute but also in chronic changes. The other aspect to toxicity is genotoxicity, where you are interested in changes to the genetic makeup. This could include something as basic as DNA damage or mutation or chromosome aberration to something that can be even more concerning, such as carcinogenicity and reproductive toxicity. And all of this also has to be contextualized against the correct animal model. So remember here, when we talk about testing from a host perspective, we are not talking about in vitro testing, although that should have been done a priori. Here we are primarily focusing on in vivo and clinical testing. And that is what is going to dictate the safety and effectiveness. In fact, in the clinical trials from phase 1 to phase 2 to phase 3 and so on, the bars for safety and effectiveness keep going up, right? So that's very important to contextualize when we are looking at things like genotoxicity, where the correct choice of the animal model is very important. You can also have toxicity arising from degradation and aging, and this is essentially once you put the biomaterial or the device into the body, the host is going to act on the material, right? And that can lead to degradation and aging. Just the presence of that foreign material in the body can have its own effects from the host perspective, as we have seen in previous lectures. Or with repeated use, especially if it's an implant that requires mechanical motion regularly, that can have or result in degradation. And just the fact that the material is going to be present in the presence of an aqueous buffer at 37 degrees can have aging effects. So all of this has to be considered when we think about toxicity. This also suggests to you the importance of site and the duration of implantation. Both of these are important. How long the implant is going to stay there, and is it going to be or result in any toxicity over that entire period, as well as the site where it is implanted. You can also have leaching of extractable components such as stabilizers or unreacted monomers, particularly in polymers. So all of that has to be tested from the host perspective. And remember that absence of toxicity is not evidence of safety. Now, safety is a much broader term, and toxicity testing that is done is typically done under controlled conditions. Even if it is in vivo or clinical, it is done under controlled conditions. And despite newer and newer standards, better standards that we have today for testing, we can't predict all the scenarios that can occur when this is eventually deployed in a patient commercially. Now, that should suggest to you that safety has many more dimensions than just toxicity. Sure, toxicity is one important aspect of safety, but the kind of toxicity testing that is done here is not absolutely conclusive of the safety of the material or the device itself, because it cannot account for all the scenarios. Although that continually keeps getting reviewed, the standards keep getting updated, etc. It's very difficult to conduct testing where all scenarios are accounted for. So there is always one caveat to be kept in mind, which is absence of toxicity in these studies, in these testing studies, which is good, is not complete evidence of safety of the medical device. Now, the other aspect that is involved in testing from a host perspective is sensitization and compatibility. Now, sensitization essentially is, we are asking a question here, which is, can a material or a chemical cause an allergic reaction after repeated exposure? So, with repeated or prolonged exposure, can this cause overreaction in a host, or what you would call hypersensitivity? And this is typically done through skin sensitization tests, topical or injection, and the animal model used is typically guinea pig. So, these could be used for some things like collagen dermal fillers to test for hypersensitivity. Compatibility testing, particularly tissue compatibility, involves histology and immunohistochemistry, where one is looking at the types of cells present and the other is looking at the expression of specific proteins at the site. Of course, this also has to be contextualized against any biochemical changes or metabolic changes that occur. So, blood draw has to be done, and you have to look at markers in the blood. You're also looking for changes like weight loss, you're looking for dietary changes, changes in sleep cycles. All of these are important and have to be contextualized against any compatibility and sensitization or any other testing that is done in vivo or in the clinic. And one thing we note here is that there are very stringent testing standards, very detailed testing standards for biological evaluation of medical devices. And one of them is listed here, which is ISO 10993, which is particularly relevant for biological evaluation of medical devices. Now, we have noted earlier that regulatory agencies such as the FDA don't mandate the use of specific standards, but if you have followed these standards, it's much easier to claim evidence of safety and effectiveness. Essentially, these standards are important because they permit standardized testing under controlled conditions, making comparisons and benchmarking much easier. So, that is why standards are extremely important. Now, now that we have looked at testing from a host perspective, we will move on to another aspect that becomes important from a host perspective for natural materials, which is the immune response. We first note that synthetic polymers do not have recognition factors such as the MHC or HLA, which means they will not be rejected like an organ transplant. Typically, what happens with synthetic polymers we have seen is they undergo the FBR, foreign body reaction. This is something we have seen in the previous lecture. So, they are not typically rejected like an organ transplant, whereas naturally derived materials have other risks because they have cells, they have proteins that could serve as antigens in the host. So, all of these factors have to be considered. So, typically they are heavily processed to remove cells, nuclear material and any non-self antigens. And two examples of xenografts that are typically used are bovine collagen and the porcine heart valve. The risks with such kinds of implantation is best understood using some case studies and specific examples. So, we will look at a quick case study, namely the decellularized xenograft transplants using porcine heart valves. So, essentially the company developed these collagen based decellularized porcine heart valves meant for patient cells to repopulate after implantation. The details of this case study are available in this reference. So, if you want more details, you can look it up. But essentially, the company that developed it, their intention was that eventually this would be a tissue engineered strategy where the patient's own cells post implantation will populate the matrix. But unfortunately, what happened is when this was in this case implanted in 4 pediatric patients, 3 of them actually died due to severe inflammation, foreign body reaction, degeneration of the valve and calcification. This was around 2001, and the 4th patient was saved only because based on the experience with these 3 patients, the surgeons prophylactically removed the valve from the 4th patient. And while there is a bunch of reasons why this happened, the primary reasons were attributed to be incomplete decellularization of the porcine heart valve, calcific deposits even before they went for implantation, and collagen that is not from human sources, xenocollagen. Now, incomplete decellularization, where if you have cell debris left, that's going to be extremely problematic from the host perspective. If you have cells from another species that remain, this is going to mount a severe immune response upon implantation. The other one, calcific deposits, we have already seen. Now would be a good time to pause and recall in lecture 32 about the issue of calcification. So, calcific deposits can make outcomes of these implants much worse. And of course, having collagen that's not from humans, essentially pig collagen for example, and when the processing is not done appropriately, you can have residual epitopes such as alpha-gal for example, that can lead to severe rejection. But do note that the same company has continued to use homografts or valves from humans. These are what are called homografts, and they have continued to perform reasonably well and they were approved to the 510K clearance, which we will look at later in this lecture through the FDA. So, it's quite clear that in this case at least, the issue came from the use of pig heart valves and not so much the human heart valves, and these were reasons that were potentially attributed to these issues. Now, segueing from here, I did mention that you can have alpha-gal like epitopes residing in these matrices, which can cause a huge risk. Now, the alpha-gal itself is essentially galactose alpha-1,3 galactose, short alpha-gal, which is an epitope found on the cell surface of most species, but is absent in humans and some other primates. It's typically found on the surface of the endothelium in most other species including pigs, and is also present in the ECM. So, you have to make the connection now to the collagen that was used here, which is one of the primary proteins found in the extracellular matrix, and any treatment processes that claim to get rid of all the antigenic material and cellular material, it's very difficult to get rid of all the alpha-gal, particularly when it is embedded in the ECM, right. So, that can cause a huge risk because it is recognized by the immune system in humans as a foreign substance through IgG and IgM antibodies. Essentially, the human body has developed these antibodies in early development. One theory is that in response to gut bacteria, they have been already developed. So, when this antigen, this epitope alpha-gal is present, residual alpha-gal is present, it can lead to something called hyperacute rejection. Essentially, this is where the body mounts a very severe immune response. This is not like FBR at all, this is much worse, leading to tissue necrosis and perhaps even death. Now, despite this risk, porcine heart valves continue to be used even today very widely in humans, and the reason is the processing has changed quite a bit. We have better methods of processing. One is glutaraldehyde fixation. So, when you have these porcine heart valves that are removed from pigs, they are fixed with glutaraldehyde and treated appropriately to mask, if you will, all of these epitopes to get rid of many of them, but whatever remaining is also masked, the collagen gets cross-linked, and that in turn reduces the immunogenicity within the host. So, once this is implanted within humans, the body does not mount as strong an immune response, right, and any immune response that is mounted is usually taken care of in the initial period at least through drugs that are supplemented. And unlike the tissue engineering strategy that this company followed here, current day porcine heart valves that are used are not considered living tissue, they largely serve a mechanical role. But because you are using GTA, and that's hypothesized to be one factor leading to calcification, you can have calcification of these valves in the long run as well as deterioration. But despite these risks, these are still widely used and they remain very popular and they perform extremely well as well in most people. And the reason is because when a patient requires a valve replacement, there are broadly two categories of valves that are recommended, either a mechanical valve or a bioprosthetic valve like the porcine heart valve. The mechanical valve is synthetic, doesn't have these risks, and is typically recommended for younger patients. But the risk or the concern there is they clot very easily. Thrombus formation is a significant concern. So, these patients, particularly younger patients who have this mechanical valve fitted in, they have to be on anticoagulant therapy for a very long time period. For such patients, the bioprosthetic valves are typically not recommended because they are more metabolically active, which means the risk of calcification is much higher, right. So, these porcine heart valves are typically prescribed for patients who are 50, 60 plus, where they will remain and function quite well for 10 to 20 years. And because calcification typically is associated with higher metabolic activity, that age group likely will not be that metabolically active. So, the risk of calcification is also lower. So, you see, it is not a white or black kind of a situation, it is sort of a risk benefit analysis. Essentially, all of these have benefits, all of these have risks. And we will again reiterate that when we look at the case study of the polypropylene mesh later in this lecture itself. But essentially, what healthcare practitioners will evaluate is the risk to benefit ratio. Overall, does this implantation the benefits of this implantation far outweigh the risks associated with the implantation, right. So, that is why in older patients typically the porcine heart valves are used quite well and they are successfully deployed as well. And any risks with or any potential concerns with immunogenicity is largely taken care of through glutaraldehyde treatments, and any potential calcification that can occur is also likely not as high as you would see in metabolically active younger patients. So, these are some aspects to keep in mind, and it is not correct to think of porcine heart valves as being completely problematic. Sure, we saw a very disturbing case study here where it led to the death of 3 pediatric patients, but what you also have to keep in mind is that for much older people, seniors particularly, porcine heart valves are regularly used and they function quite well. So, if you are still concerned that these are risks, then there are alternatives. These are developed more recently. One is you can have enzymes that can remove selectively these epitopes. Alpha-gal is just one of them, there are others as well. Or more recently you've had knockout pigs, essentially pigs that are genetically modified to not produce the enzyme that results in the formation of alpha-gal. So, if you do not have the enzyme to produce alpha-gal, alpha-gal is not going to be produced, which means there is no risk of alpha-gal related hyperacute rejection. So, some quick points to keep in mind in terms of newer ways of dealing with the alpha-gal risk. Related point to consider is this idea of hypersensitivity. Now, it is nowhere close to the kind of risks and severity that is usually seen with hyperacute rejection, but hypersensitivity can typically occur with bovine collagen dermal fillers. So, hypersensitivity essentially can be of different types. Typically with these kinds of things you see either a type 1 or a type 4 kind of a hypersensitivity. Type 1 will be very quick and rapid, you will see redness, itching, swelling, etc. Type 4 will have a slightly later onset and can even result in nodules. Essentially they are all some kind of an allergic reaction, if you will, that the body is mounting in response to bovine collagen. And typically these are taken care of through oral antihistamine and corticosteroid supplementation, or in severe cases particularly type 4, if you have nodules etc., the corticosteroid can be injected at the site, and usually this subsides with time as the body gets used to the filler. But one way to avoid this, if you are concerned or if the patient is extremely sensitive, is the use of hyaluronic acid filler, which again provide viscoelastic supplementation without the risk associated with bovine collagen dermal fillers of hypersensitivity. So, that leads us to more severe issues that could potentially be noticed with implantation of medical devices. Essentially, we are looking at adverse responses and retrieval. Keeping in mind that the goals of implant retrieval and evaluation are typically to determine the rates, modes and mechanisms of failure. Now, when we say failure here, this does not necessarily mean death. The implant could have failed over time just because it has been around for the porcine heart valve, for example. With time it can deteriorate. But if that risk is known in advance, then they can be taken out before they completely fail, and the patient can be fitted with a new valve. So, failure here is sort of a broad term, and that is how we are using that word here. The goal here is to identify the effects of patient and implant factors, very important, both patient and implant. So, essentially host biomaterial interactions. And remember that this is a two-way arrow, never forget that. This is something we have seen repeatedly throughout the course. Biocompatibility is a two-way street. The biomaterial, sure, or the device is going to have an effect on the host, but the host in turn is going to cause changes to the biomaterial, right from aging to deterioration to calcification to FBR to remodeling, all kinds of things that we have seen in the last few lectures. All of that is going to dictate the performance and clinical outcome of these medical devices. And the other thing that should be considered as far as evaluation of failure is concerned is any temporal variations in mechanisms of both tissue biomaterial and blood biomaterial interactions. We spent an entire lecture, lecture number 32, looking at blood biomaterial interactions, particularly thrombus formation. So, all of this has to be continually monitored. That is what temporal variations mean, because the host is going to continually act on the implant or the medical device. The medical device is going to exert its function. So, these properties can change with time. So, this is why we said safety is a very loaded, it's a pregnant word. There is a lot of dimensions to that word safety, particularly when you now factor in the temporal variations. Safety takes on a much larger dimension. Now, some adverse effects that could be seen or some adverse interactions within the host include thrombotic occlusion or thromboembolism, which has to do with blood material interactions. This can lead to severe clotting, leading to obstruction in the blood flow. And remember that homeostasis is a delicate balance between factors that promote clotting and factors that promote bleeding. So, either of these extremes is bad, right? So, and here we are looking at the scenario where you have excessive clotting or thrombus formation. You can also have anticoagulant therapy related hemorrhage. So, when I said that patients, younger patients who are fitted with mechanical heart valves, they have to be on warfarin like medication for the rest of their lives to prevent clotting, which is a known risk with mechanical heart valves. But if you are systemically taking anticoagulant drugs, blood thinners as they are called, there is a risk associated with hemorrhage, because this can have systemic effects. And so, that also has to be accounted for. That is one kind of an adverse response that can be noted with some of these anticoagulation therapies. You can also have embolization or lymphatic spread of fragments, and this could simply be debris coming from wear of the material with time. You can have incomplete healing or excessive tissue growth. One example of excessive tissue growth is very thick fibrous capsule formation that leads to isolation of device, right. So, FBR per se is not a problem, it is the severity of the FBR and the thickness of the fibrous capsule, as we saw in the last lecture. That is what dictates the eventual success or failure of an implant. So, a very thick fibrous capsule is not with a lot of foreign body giant cells is not good at all. Of course, you can have material wear, fracture, fatigue and calcification. Now, some of these are coming from repeated use in the patient, some of these could come because of blood material interactions such as calcification. Of course, there are other aspects to look at, which is inflammation, which we have looked at in the last lecture in quite some detail, as well as allergy, toxicity and tumor formation. Some of those aspects we have looked at in this lecture. So, these are all examples of adverse interactions that can occur within the host. Now, to drive home this point about implant retrieval, analysis, etc., let us take a quick look at a very important case study of the use of polypropylene meshes for pelvic organ, for correcting pelvic organ prolapse, in short POP. So, essentially polypropylene meshes, if you recall, we have seen earlier that they are used quite a bit for hernia repair. And what was done in this case was pelvic organ prolapse is essentially a condition where organs in the pelvic region start to descend, and typically a mesh is used in essentially a transvaginal mesh to treat POP. But what happened here is, because polypropylene was used, there is a significant mismatch between the mechanical properties of the stiff polypropylene mesh and the sensitive tissue under consideration here. So, what happened is, normal bodily movements led to the mesh deforming, stiffening and contracting, leading to pain, erosion and nerve damage for the patients. Patients also reported chronic inflammation, oxidative degradation and particle shedding. Now, what happened here? Why did this happen? And what are the lessons we can learn from this case study? Essentially, the first lesson to be learned from a design perspective is this viscoelastic mismatch. While polypropylene meshes are approved for hernia repair, note that the site of implantation there is the rigid abdominal wall. So, there is no issue there. The issue comes here because the mismatch happens between the viscoelastic or the dynamic and sensitive vaginal microenvironment versus the stiff polypropylene meshes. So, what works in one region does not work in another region. There is also the issue of stability that has to be considered. Polypropylene is considered quite stable, but you see that in vivo microenvironment can induce erosion over years. This is why I said safety. This is a very different kind of a dimension to safety that we are seeing here now, right? This could have worked very well in a different application such as hernia repair, or may have even shown reasonable outcomes for short term implantation, but this is long term implantation, and over years the in vivo microenvironment can induce erosion. That is what caused chronic inflammation, particle shedding, etc. in this case, leading to a lot of pain for patients and irritation. Now, these two have to do with the material itself. There is one more aspect to be considered here, which is another lesson to learn, which is the mode of implantation. The mode here was the transvaginal mode. There is also the transabdominal mode. The issues noted with the transvaginal implantation were so severe that in 2016 or so, the FDA had to reclassify this as a class 3 device, and what class 3 is, etc., we will look at very shortly in this lecture itself. And in 2019 or so, I think it was completely banned, at least in the US, by the FDA for treating pelvic organ prolapse, the transvaginal route. Transabdominal doesn't make it any better as far as the mesh itself is concerned. So, the mesh issues continue to persist, but because the risks associated with transabdominal implantation are much lesser compared to transvaginal, transabdominal is still used in cases where again the healthcare practitioner will determine that the benefits will far outweigh the risks. Now, what we will look at is two quick examples of case studies where some unusual issues can occur because of storage and handling issues. Essentially, you can have either inappropriate storage conditions leading to performance issues in the implant or the device, or you can have handling issues during implantation. These at face value may not seem like they are significant issues, but they can have very severe consequences for the performance of the material or the device. So, we will first look at the performance or the issues associated with storage and its effects on performance. So, we are looking at catheter material, and essentially we are looking at polymer catheters made from polyether block amide elastomers called PBA for short. These are used and can become brittle from exposure to strong disinfectants or UV light during storage or cleaning. That leads to accelerated degradation and oxidation of chains, which reduces the material's mechanical properties, particularly tensile strength and toughness. So, you see here, this intrinsically this material is not problematic, but when it is exposed to strong disinfectants or UV light during storage or handling, or perhaps even some extreme sterilization conditions, that can lead to oxidation of chains and accelerated degradation, thus significantly negatively impacting the mechanical properties of the catheter itself. So, the lessons to be learnt from this one are the service condition compatibility. Essentially, manufacturers must understand and specify the entire life of a product, including cleaning and storage, and not just from the time it leaves the factory, but all the way from the time it is shipped to the site of use, the storage conditions that must be adhered to and the precautions that must be taken therein until the point of implantation and surveillance thereafter. So, manufacturers should understand and specify the entire service life of a product, right from the time it leaves to the time it is implanted and used, as well as some education in hospitals, right. So, this failure may occur due to external handling and chemical environment of the hospital. So, clinicians and people handling these things should be educated. So, this is one sort of an unusual kind of a case study where inappropriate storage conditions can lead to degradation, thus affecting the performance. You can also have similar issues occurring due to inappropriate handling. We have already seen that hydrophilic polymer coatings are used on catheters. This is again a catheter example for minimally invasive procedures to ensure easier insertion into the blood vessels. So, hydrophilic polymers like PVP are used and coated on these blood vessels, these catheters, that is going to aid easier insertion within the blood vessels. Now, if the coating is inadequate or if the catheter is used inappropriately or improperly, these gel fragments from the coating can peel off and travel through the bloodstream as emboli, and that can be severely problematic because it can lead to organ accumulation and potentially even death. So, these fragments that dislodge from the coating can lead to such severe issues. So, again the lessons here from the manufacturer's perspective or from a biomaterial design perspective, the lesson is can we come up with better coating adhesion and better stability. So, there is a need for robust adhesion between coating and the base polymer substrate, and newer ways to stabilize that and to make it very adherent have to be consistently looked at. From a clinician or a user perspective, this knowledge about these device fragments potentially becoming foreign bodies and causing emboli is also important to be kept in mind. So, again just two quick examples of some unusual case studies that can occur during inappropriate storage and handling. Now, we will move on to another very important or two back-to-back case studies that are very important. One is the Dakin Shield intrauterine device case study. The other is the Vitek implant case study, which is not unfamiliar to us, which we will look at on the next slide. Now, before we start looking at the Dakin Shield intrauterine device case study, we will first take a quick look at what IUDs are. These are essentially IUDs. Essentially, they are used for reversible female contraception, and they are typically implanted and they have different mechanisms through which they act, particularly inducing some kind of a mild chronic inflammatory microenvironment within the uterus, either to prevent fertilization and or implantation of fertilized egg in case fertilization occurs. Now, this device has tail strings attached to the distal end to facilitate easy removal, and this has to be implanted by a trained clinical practitioner. There are both copper and hormone based systems that are used, and the mechanisms by which they act and prevent pregnancy are slightly different. We are not going to get into the details of that for this case study, but it is good to know that there are different ways through which this can act. And in today's world, modern day IUDs involve a polymeric design where you have a T-shaped body made from LDPE, this kind of a T-shaped body, where barium sulfate is typically added for opacity. Essentially, this will show up on an X-ray if barium sulfate is added, and coiled copper wire with at least a minimum 99% purity is used. And please note these specifications, because it turns out that some of these specifications will become important for why some of the older designs, particularly the darkened shield IUD, failed. So, with mandated surface areas between 200 to 380 millimeter square, and the tail string, like so, as shown here, are highly drawn and oriented polyethylene or polypropylene monofilament strings. This word monofilament is very important. Why that is important will become clear in just a minute. And there are guidelines for what kind of tensile strength it should have to withstand the kind of tensile load. Again, these are things that we have seen when we did mechanical testing of biomedical devices and implants. So, it will be good to recall some of our concepts from tensile testing. Now, this is as the IUDs stand today, but if you go back in time and revisit the 1960s darkened shield IUD, its failure is often cited as the reason for prompting the Medical Device Amendments Act of 1976, which is something we have looked at in lecture number 5 when we discussed the Vitek implant case study. So, now would be a good time to pause and recall, or if you are not familiar with this, I would suggest looking up the Medical Device Amendments Act of 1976 in the United States. Essentially, this IUD, the darkened shield IUD, had a design with a nylon multifilament tail strings. Note that this is a multifilament as opposed to the current standard, which is a monofilament. That small difference, as it turns out, will become a very key factor in the failure of these darkened shield IUDs. Essentially, it is a design with inner fibers encased in a smooth outer sheath, nylon, and the manufacturer actually claimed that the thinner membrane in the IUD could lead to higher surface area, and they had these fins, plastic fins, that could promote better attachment. So, they had claimed that it was actually a better design, but it turns out, unfortunately, that this led to pelvic infections, miscarriages and deaths. What went wrong? This multifilament tail, it turns out, was one of the primary culprits. It was without the heat treated ends, it led to wicking of the vaginal fluid and provided bacterial access into the uterus. So, because this had this multifilament kind of a tail string design, now this bacteria from the outside could sort of piggyback and wick and go in to capillary action and reach the uterus, which would otherwise not be allowed, because the cervix has a self-cleaning mechanism, and that was sort of surmounted by the bacteria being able to now go through the multifilament tail strings through wicking or through capillary action, right? And also deterioration of the outer nylon sheath was another factor, which also provided a route, again, like I said, for the bacteria to enter the uterine cavity. And although the manufacturer had claimed that this was a better design, it also led to a lot of pain for the patients, and string breakage is also noted during removal. So you see that one small change going from monofilament to multifilament had such a large impact on the performance of the IoT, leading to very stringent guidelines for tail strengths, and particularly with sufficient tensile strength to avoid breakage. That's going to address this issue. Of course, the second criterion is biological inertness to prevent changes during use, which is anyway very important. And the third is the requirement for a hydrophobic monofilament fiber to avoid this capillary action. So that's going to address this issue, right? That's why I said that the word monofilament here is very important. Current designs mandate this. So you see, this sort of tells you how the design of biomaterials and medical devices has to be carefully taken into consideration from all aspects. So this was not something that the company would have anticipated, and yet this is what happened, right? And that led to a lot of issues for patients, and like we noted, even deaths, right? So that is one very important landmark case study for failure. The other one is the Vitek implant case study. We are simply revisiting it here. We have looked at it in very great detail in lecture number 5 when we first started looking at the use of polymeric biomaterials and why structure property function relationships are important to understand in that context. So we are just going to recapitulate some key aspects here. So this company, the scientist who founded this company was proposing the use of these Proplast based implants, essentially porous PTFE based implants, for correcting dysfunctional temporomandibular joint discs issues, right? So when patients have dysfunctional TMJ discs, then this was one of the devices that was suggested. Essentially, it was a Teflon FEP resin laminated with a porous PTFE. The key here is the porous PTFE, and the hypothesis at that point was that the porosity could encourage cell infiltration and could promote osseointegration, right? So porosity, as we have seen, is one of the factors that can dictate FBR, right? So we have seen that smooth, non-porous materials typically have a more severe FBR. So at least on the face of it, it might appear that the porous PTFE might actually be better. In fact, it might even serve to promote cell infiltration and promote osseointegration. What they didn't account for was the site of implantation, the mechanical forces that would be involved there. Chanley, if you remember, had already warned against the use of PTFE in certain applications because of its poor wear properties. And in this case, what happened is once they were implanted and used, it led to a lot of wear debris because of mechanical motion, excessive chronic inflammation, very severe FBR, and the presence of a lot of FBGCs around the implant. So shown here is a quick timeline capturing all those events. What we also want to note here is how did this get approved at all in the first place, right? So you see that the 1976 FDA institutes the Medical Device Amendment Act that I talked about in the previous slide, and in 1978 is the earliest known recipient of this Vitek implant device. And you notice that when they first started using it, right, especially with initial clinical studies, etc., and it was marketed eventually, FDA grants it 510K status. Now 510K is something that we have alluded to briefly earlier, and we will look at in far more detail on the next couple of slides when we look at regulatory agencies and regulation of medical devices through the FDA. Essentially, this is a fast tracking procedure to approve or clear, not approve, but clear medical devices for use based on substantial equivalence to a previous device. And what this person did was to claim substantial equivalence to a silicone sheeting that was used for a similar application. And you can already see, if you've understood structure property function relationships quite well, you can see that silicone is a very different kind of material, right? So claiming substantial equivalence is incorrect, and also the company had not done any detailed clinical trials, And any animal trials that had been done were also inappropriate, right? They were alveolar ridge reconstruction studies, which is quite different from the TMJ reconstruction. So, a bunch of things had gone wrong here. Anyway, so ultimately the animal studies performed later showed catastrophic results. Note that this was after clinical deployment, which is sort of very, very bizarre. Anyway, so ultimately this the FDA comes to know of it and then reclassifies it as a class 3 device and eventually withdraws all the implants from the market. Okay, that's what happens here. And just to drive home that point about severe FBR, we'll also look at a H&E stain histological section of the implant showing acellular foreign body material adjacent to reactive giant cells. So, that is what is shown here. So, this is severe FBR, and if you remember the two hallmarks of FBR are, can you recall what they are? Very severe FBR is associated with two very important hallmarks. One is thick fibrous capsule, second is lot of FBGC activity. Excessive presence and continued activity, which is what led to chronic inflammation, right? And the fact that this was sending out debris over time because of wear was only making things worse. So, this is another reason for us to now look at how regulation is done. Both the darkened shield and the biotech implant case study, one thing we can learn is that medical devices have to be regulated very carefully. So, we will take a quick look at regulatory affairs for medical devices, and we will first note that we can't talk about one generic approval pathway. It's country specific, and what is approved by one is not automatically approved by the other. So, shown here are examples of agencies in different countries: the CDSCO in India, FDA in the US, EMA in Europe. Then you have similar agencies for Brazil, Japan, etc. So, instead of looking at a generic pathway, what we will do is focus on the FDA as an example, and I will give you a 2-slide summary of a very complex approval process pathway. And the hope is that when you come across similar approval workflows, etc., for other regulatory agencies, it will be easy for you to contextualize. So, with those two caveats in mind, we will quickly take a look at the FDA's approval pathway for medical devices. Firstly, we note that the FDA is a law enforcement agency. It can cite, fine and prosecute for violations, and it provides approval for drugs, biologics and devices. And it turns out that there are multiple wings or branches within the FDA, and the center responsible for devices, medical devices, which is the primary focus of our lecture here, is CDRH, Center for Devices and Radiological Health. Essentially, it's the branch responsible for regulating medical devices. Of course, you can have others like CBER, CDER, one is for drugs, biologics, etc. So, these are other wings that look at drugs, pharmacological agents, immunotherapies, vaccines and so on, which are not covered under medical devices. Of course, if sometimes when two are involved, such as drug-eluting stent, it becomes a combination product. Now, for the purposes of the regulatory agency itself, a medical device is defined as an instrument, apparatus, implant, machine, in vitro agent or similar related article that is used to diagnose, cure, prevent or treat diseases or other conditions, and as we have noted earlier on one of the earlier slides in this lecture, is intended to affect the structure and function of the body primarily through mechanical or physical means. It does not achieve its effects through chemical action or is metabolized. Essentially, it is not a drug or a biologic. So, these would be regulated through CBR, ER or CDER depending on the type of therapy. So, that is quite different. Now, the approval pathway, it turns out, is based on a 3-tier class based approval depending on the risk that the medical device poses. So, shown here is a bird's eye view of the approval pathway for the product. It's also available for the company, which we're not going to focus here. For example, this will require specific GMPs, good manufacturing practices, which is also covered under this pathway for medical devices. We're not going to focus on the company here. Our goal here is to look at the product. And like I said, there's a 3-tier classification based on risk of the product. And higher the risk, higher is the evidentiary threshold required for safety and effectiveness. So remember, these are the two pillars, as we have already said, that agencies such as the FDA will evaluate, approve and regulate medical devices. And class 1 is typically low risk. It requires only FDA product registration, and some examples include arm slings, bedpans, etc. Class 2 is a moderate risk medical device, which requires a pre-market notification, in short PMN, and this is also the 510K pathway that we have continuously alluded to throughout this lecture, details of which we will look at on the next slide. Examples that are cleared through this include catheters, powered wheelchairs, etc. And of course, there is the class 3 high risk category, which requires pre-market approval or PMA, which specifically involves clinical trials, and examples include pacemakers, total joint replacements, etc. And of course, if you have combination products, this could include drugs, devices and or biologics, and we saw that drug-eluting stent, because its primary action is that of a device, is considered a device-led combination product. So, it will primarily be regulated by, for example, the CDRH, with of course appropriate inputs from CDER, so that they can have the drug aspect also taken care of. Now, that is the overall picture of the approval pathway. Now, we will focus a little bit more on both class 2 and class 3 devices, particularly with reference to the 510K pathway as well as the pre-market approval pathway. One is just clearance, the other is approval. So, first we look at the pre-market notification pathway, which is the 510K clearance. This is typically used for class 2 devices, and essentially what the manufacturer is expected to show is substantial equivalence to an approved predicate device in terms of both safety and effectiveness. So, this is sort of a fast track pathway that manufacturers can take based on the risk, both in terms of safety and effectiveness, if they can show substantial equivalence of their intended product or device to a predicate device, both in terms of safety and efficacy. So, FDA has specific guidelines for that. So, it has to be either the same intended use and the same technological features, or the same intended use and different technological features as the predicate device, but without raising concerns of safety and effectiveness. So, either it can have the same intended use and the same technological features as the predicate device, which has already been approved, or it can have the same intended use but different technological features, but this should not raise any concerns of safety and effectiveness, and that documentation has to be separately submitted to the FDA, and that should be backed by evidence. And typically for these kinds of approval pathways, very standard testing methodologies are available through ASTM or ISO, and they are very highly recommended. As we have already seen, standardized testing is not mandated by the FDA, but if you use these standardized tests, it is almost always expected that you will use standardized tests. So, that is sort of the expectation. As long as you are using a particular standardized test that is well established, you should be okay. Because it is sort of a fast tracking kind of a scheme, lab effectiveness and biocompatibility are required, but you typically do not need clinical trials for these kinds of approvals, especially the 510K clearance, because you are claiming substantial equivalence to a predicate device. And any device that comes out of this pathway successfully is called FDA cleared. This is not FDA approved. So, you see that there is a very big difference to what is FDA cleared versus what is FDA approved. They are not the same thing. FDA cleared can still be used commercially. Manufacturers can deploy it commercially, clinicians can use it commercially, except that it's the technical word, it turns out, is not FDA approved, it's FDA cleared. And this typically does not mandate a formal post-market surveillance plan for the device. We have seen examples of some of these devices that have been used in this lecture, some inappropriate use, particularly for example the transvaginal polypropylene mesh. I said that the FDA eventually had to classify it as a high risk device, class 3 device, right. Originally it was fast tracked through the 510(k) clearance, which was moderate risk, right. Likewise, we saw the Biotech implant also had claimed substantial equivalence to a predicate device and was classified initially as a class 2 device, went through 510(k) clearance, and eventually because of the issues noted, it had to be reclassified as a class 3 high risk device. While those are all inappropriate abuse of the 510(k) clearance pathway, the 510(k) clearance pathway is not per se a villain. That's definitely not the impression I want to give to you. It's a very powerful pathway. If all medical devices had to go through the full pre-market approval pathway, which is what we're going to look at next, with extensive clinical trials, etc., many of these therapies and devices will take a very long time to reach patients. So, there have been a lot of success stories that have gone through the 510(k) clearance. What we are looking at here is very specific cherry-picked examples, but nevertheless very dramatic examples, very devastating examples of abuse of the 510(k) clearance, right. So, the pathway itself isn't problematic. It's the abuse and the inappropriate use of the pathway, incorrectly claiming substantial evidence to a predicate device, that is problematic, right. So, that distinction is good to make, that the pathway itself has led to a lot of successes that have reached patients very rapidly. So, it's a fast tracked pathway, but again one has to be cautious against these pitfalls that we have noted on these previous slides with these these unfortunate case studies. Again, as far as testing and standards are concerned, now would be a good time to pause and recall that when we did mechanical testing in lectures 22 and 23, we looked at several examples of standards that are typically used. I would also give you a specific example here, and I would suggest that you look up the standards used for testing this X repair, which is essentially a woven PLLA mesh used to augment surgical repair of tendons and soft tissues. Essentially, this has gone through and been cleared by the 510(k) pathway, and it's been successfully used. So, you can take a look at some of the standards that this company has used for clearing this as a class 2 device for augmenting surgical repair of tendons and soft tissues. In contrast, the pre-market approval is typically used for class 3 devices which carries a higher risk. So, the standards are stricter in that sense, and the class 3 and sometimes some higher risk class 2 devices require clinical trials and pre-market approval. So, this you can't claim substantial equivalence to a predicate device. This has to go through the full round of clinical trials and has to have pre-market approval, which is PMA, whereas the 510(k) clearance is the pre-market notification. They are not the same. And typically with these kinds of devices, class 3 devices which are high risk, and some higher risk category class 2 devices will have to go through pre-market approval, which is the full round of clinical trials, and any device that comes out successfully from this pathway is called FDA approved. So, the 510(k) clearance is called the FDA cleared, whereas the pre-market approval pathway, a device that comes out of it successfully, is called FDA approved. So, when you look at devices, pay very careful attention to whether it's FDA cleared or FDA approved. They are not the same and cannot be used interchangeably. FDA approved also requires formal post-market surveillance. That's a mandatory requirement, whereas in the 510(k) pathway, although medical reporting is of course done post implantation and post clinical deployment, the FDA itself does not mandate a formal post-market surveillance plan, although after deployment they can ask for specific types of reporting, whereas in the pre-market approval situation, the FDA approved device has to have a formal plan in place for post-market surveillance, which includes manufacturers to report adverse events, sensitivity, side effects, malfunctioning and serious injuries not addressed in the device labeling or not presented in the pre-market approval documentation. Of course, doctors are also expected to report deaths. Patients can voluntarily report issues that they face. So, all of that is included within post-market surveillance and reporting. One quick aside point about off-label use, because we are looking at devices. Medical practitioners are legally allowed to use their judgment for the use of devices or formulations in conditions where the said device or drug is not specifically approved for that indication. So, in many times this is typically seen in many cases with drugs, for example, right. So, when drugs are approved for certain specific indications, clinicians in their vast experience treating many patients may consider legally the use of those drugs in other indications where the original approval didn't exist for that kind of an indication or condition, right. So, that is what we are saying here. This is what is called off-label use. So, the label itself will not specify that this device or this medicine or this tablet, etc., or this drug can be used for that particular indication or condition. So, the clinicians use their judgment and vast clinical experience for prescribing off-label use. Now, that brings us to the end of this lecture and the course as well, and I will offer some concluding remarks. We will first take one last look at biomaterials. So, this is a slightly more updated definition, which is just a material designed to take a form that can direct through interactions with living systems. So, this can be bioactive, the course of any therapeutic or diagnostic procedure. So, it still retains the essence of the older definitions, but is now updated for newer and newer therapies and devices that come out. It still retains the flavor of the diagnostic and therapeutic components, and it can now proactively engage with the host through interactions with living systems. Likewise, we will take one last look at our familiar good old friend biocompatibility. This is a word that we have seen extensively throughout this course, and perhaps now it is clear to you why this lecture was also titled Biocompatibility, Medical Devices, Translation and Regulation, because they are all deeply connected. And we are taking one last look at biocompatibility here, which is a definition from again David Williams, which refers to the ability of a material to perform its desired function with respect to a medical therapy without eliciting any undesirable local or systemic effects in the recipient or beneficiary of that therapy, but generating the most appropriate beneficial cellular or tissue response in that specific situation and optimizing the clinically relevant performance of that therapy. So, this also now you see encompasses definitions of tissue engineered products. Right, and always remember, this definition still includes the idea that biocompatibility is intrinsically a system property, and this system includes biomaterial and a host, right. This ability of a biomaterial to perform its desired function with respect to medical therapy is coming at it from the biomaterial perspective without eliciting any undesirable local or systemic effects is coming from a host perspective, but generating the most appropriate beneficial cellular response or tissue response in that specific situation is a combination of both, right. It involves both properties, both biomaterial and host property. So, it is a very nice, very beautiful definition that encompasses all different dimensions or perspectives to biocompatibility. This is the title of a leading opinion paper from David Williams, published in the journal Biomaterials in 2014. What this does is it tells you that it's incorrect to think of a specific material as being universally biocompatible, right. It's very contextual, as we have seen repeatedly throughout this course, and this has to be connected to this definition of biocompatibility. And in closing, we will take one last look at biomaterial design, which now consolidates all the aspects that we have seen throughout this course, right, from structure properties affecting function and performance, to solvent and temperature effects within the host, which affect biomaterial performance. They are also important in processing of polymeric biomaterials. Mechanical properties in microstructure affecting the performance and host responses. Surface properties and functionalization, which we've also looked at right from protein adsorption to blood compatibility to eventually performance in a host. From a material host interaction perspective, you can have degradation, aging, wear and leachables. And from a more translational perspective, you can think about processing, sterilization and manufacturability. And from a more regulatory perspective, you can think of the regulatory requirements required and the clinical deployment thereof. And of course, you're always interested from a host performance in safety and FBR or effectiveness, and also long-term compatibility and lifetime performance. So, long-term compatibility has to do with safety, lifetime performance has to do with effectiveness, right. So, that's in one short set of bullet points, we've covered all the key aspects that we have looked at throughout this course. Now, what's next on the biomaterials horizon? One can make predictions for the next 50 years, 100 years, etc. But in the words of the inimitable Neil deGrasse Tyson, human beings make lousy predictions about the future. And he gives some very interesting examples of how predictions were made earlier about different aspects of life and how all of that was completely wrong. So, I will not venture any guesses for where the field is going to go. But if you have been intrigued by all the concepts you have learned in this course, the possibilities are very, very exciting. And in closing, I will leave you with one thought again that sort of is a take home from the title of the course itself, which is always remember that structure affects property. Both of these are affected by processing of materials, and all of this together affects the function and performance of polymeric biomaterials in a host.